Hello! So today I'm going to be presenting about the gut microbiome lab, which was done by me, Courtney Sakota, Morgan Lilly, Elena Whipple, and Jocelyn Ramirez. Introduction, I just want to talk a little bit about what the gut microbiome is. So the gut microbiome is a home for bacteria and microorganisms in the gut of an animal. It correlates to animal health, and different diseases affect the microbiome, as well as diet, medication, environment, etc. So some of the links we've found with the gut microbiome is in Down syndrome. So individuals with Down syndrome show relations in their gut microbiomes in individuals. There's an increase of Parasporobacterium and Centrella in the gut microbiome. This also correlates to the aberrant behavior checklist, which allows for the conclusion that the microbiome have features and affect Down syndrome people. We saw that this experiment was done with 17 Down syndrome patients. We also found a link to gut microbiomes and obesity. So individuals with obesity show relations. In a study conducted by researchers, they analyzed blood plasma and stool samples from 674 participants and found 19 metabolites that could be linked to the person's BMI. The biggest and most important being glutamate, and it had the strongest correlation to obesity. So it just shows us a little bit of how we can see the gut microbiome in everyday health problems. The question that we were testing here was, is there a relation between the gut microbiomes of certain organisms? And the hypothesis that we decided to test was that there will be a relation in the gut microbiome of dogs with epilepsy that will differ from the gut microbiome of dogs with decreased kidney function, and the dogs with decreased kidney function will have a similar gut microbiome. The samples we decided to use to test our hypothesis were from 4 dogs. We used sample 1055, which was a female dog. She is a toy miniature poodle, about 13 years old. She weighs 12.1 pounds. She eats Trader Joe's dog food and wet food. She's been eating this for about 2 to 3 years. She has no other cohabitating animals that live with her, and she has epilepsy. The next sample is sample 598, who's a female and a yellow lab. She's about 14 years and 5 months old. She weighs 75 pounds. She eats veterinarian kidney assistance food and has been eating that for about 8 months. She lives with 2 other dogs, and she has kidney disease. The next sample is sample 855, who's a male Irish Terrier. He's 4 years old. He weighs 50 pounds. They don't tell us what diet he has, but he's been on the same diet for about 2 years. He lives with a cat, and he has epilepsy. And sample 702 is a female Ihasa Apso mix. She's 17 years old. She weighs 21.5 pounds. She has decreased eyesight. She eats low protein and various brands and has been eating that for about 3 years. She lives with 1 other dog, and she has decreased kidney function. I just want to talk a little bit about the protocol. So as a brief overview, we had 3 major steps in this experiment. First, we isolated the DNA from the fecal samples using a kit called the Quiagen kit, which is specifically designed for isolation of DNA from stool samples. Second step we did was quantify the DNA using a UV absorbance spectrophotometer, and we normalized the DNA samples. And then lastly, we used qPCR to quantitatively amplify a specific region of the bacterial genome using a group of specific primers to determine the proportions of these groups in the stool sample. To go into a little bit more depth about the procedures, for purification of the DNA from the fecal matter, we use the Quiagen spin columns. So first we used 700 microliters was added to the spin column. We closed the cap and we centrifuged it for 1 minute at 14,000 RPMs. We discarded the filtrate, which is the liquid that flowed to the bottom of the tube. We carefully opened the Quiagen spin column and added 500 microliters of the wash buffer AW1, centrifuged for another minute at 14,000 RPMs, and discarded the filtrate at the lower tube. Then we carefully opened the Quiagen spin column, added 500 microliters of wash buffer AW2, centrifuged for 3 minutes at 14,000 RPMs and discarded the filtrate. And then for step 4, we centrifuged for 3 minutes to dry to make sure that no wash was still left in the column that would interfere with the future steps. And then we transferred the spin column into a new 1.5 microcentrifuge tube, discarded the old tube. Then we micropipetted 200 microliters of buffer ATE directly into the spin column, incubated for 1 minute at room temperature, centrifuged for 1 minute to elute the DNA, remove the column, and then put the spin column on ice. And then as far as the qPCR setup, why did we choose qPCR? We chose it because compared to PCR, it's quantitative and it quantitates the amount of product it started with. So we used 2 primers. We used the forward and reverse primer that binds the target sequence before the DNA polymerase can bind and start synthesizing the DNA. The positive controls received the forward and reverse primers, and so did the negative. And then why did we use Cyber Green? We used it because it binds to double-stranded DNA, but not single-stranded DNA, so little fluorescence is emitted into Cyber solution. It's simple and cost-saving, and the signal intensities correlate with DNA amplified, so the initial sample input amounts. When looking at our data, the 260 to 280 ratios, we had for sample 1055, it was 1.82. For 598, it was 1.4. For 855, it was 1.81. And for 702, it was 1.45. And up here, we mentioned that the ratio is a measure of purity and should be approximately 1.8. So 2 of our samples, both of the dogs with epilepsy had a 1.82 and a 1.81. And then the 2 dogs with kidney disease had a 1.4 and a 1.45, which showed a presence of contamination. But it kind of goes along with our results because the kidney disease dogs had lower, and that shows that they aren't able to process proteins. And when you have kidney disease, protein processing goes down, so it makes sense and it aligns with our data. And then for the first set of forward and reverse primers, we did bacteria and fungi. So for our sample, for our positive control, we got a 17.08 CQ value. Negative control, we got a 27.20. Dog with epilepsy, 1055, we got a 21.70. 598, we didn't get a value. And then 855, we got 25.77. And then for our fungi results, we got a positive control of 34.45, a negative control of 36.95. Sample 1055 had a result of 36.17, 598 had a result of 34.21, and 855 had a result of 34.20. So then for our bacteria to fungi ratios, this one, for our positive control, it was 169.7. For our negative control was 86.42. This one was 22.7. And then this one was 344.49. Next set of data, we did actinobacteria and firmicutes. So for our actinobacteria and firmicutes, we did not do a positive control, we just did a negative control. So for our negative control with actino, we got 31.71. For dog with epilepsy, we got 1055, we got 23.99. For sample 598, we got 37.62. Sample 855, we got 31.75. And sample 702, we got 27.30. In firmicutes, our negative control, we didn't get a value. Our sample 1055, we got 29.35. We didn't get a value for sample 598. We got 23.36 for sample 855. And for sample 702, we got 23.24. As for our ratios, there was no ratio for negative control because we didn't get a value for firmicutes. For 1055, we got 41.23. For 598, we didn't get a ratio for the same reason as negative control. For sample 855, we got a ratio of 0.003, and for sample 702, we got a ratio of 0.0598. And for our last set of data, we did bacteriodete and microbiome. For a negative control, we got 31.15. For sample 105, oh, this is with the bacteroidetes. For sample 1055, we got 31.06. For 598, we got 34.12. For 855, we got 26.07. And for 702, we got 21.12. And for the varicue microbia, we didn't get any results. All of them came up NA, so we don't have any ratios for that. I just want to wrap up with a little bit of discussion about our data and our experiment overall. So the extraction of our DNA was not very successful in terms of quantity. In each sample, the DNA was either rare or essentially absent. So in order to determine that, the concentration was supposed to be below 20 to be considered abundant, between 20 and 25 to be rare, and above 25 was essentially absent. So if you look at just a few of our data points, it's clear that the DNA extraction quantities were unsuccessful. For example, when you look at bacteroidetes, sample 1055 had a concentration of 31.06, and sample 598 had a concentration of 34.12. That's not the only sample that the quantity was unsuccessful. When looking at actinobacteria, the sample 598 had a CQ value of 37.62. So our values were very high, and they were pretty much considered essentially absent. So we didn't get very much DNA when we did our extraction. In terms of quality, our DNA was pretty successful. All of the values were in the good quality range for the 260 to 280 ratios. We got values of 1.82, 1.81, 1.45, and 1.4. And the samples that had the lower numbers were the ones that had kidney disease, so it makes sense why their numbers were a little bit lower than the dogs with epilepsy. So we did have relatively good quality DNA, it's just our quantities were lacking. All right, so the negative controls in the first round of qPCR was 27.20 in bacteria and 36.95 in fungi. The positive control in bacteria was 17.08, and in fungi it was 34.45. The negative controls were high this round, which does prove that there was activity and no contamination, but the positive controls are not super consistent. One is really high at 34.45, and the other is really low at 17.08. So it doesn't really tell us a lot about that set of data. So in our second run of qPCR, the negative control was 31.71 in actinobacteria, and we did not get a value in firmicutes. So the negative control here is again high, but we did have some values that were close to and some that exceeded the negative control. For example, we had values at 23.99, 37.62, 31.75, and 27.30. So if the negative control was 31.71, and we had some values like 31.75 and 37.62 that exceeded the negative control, it does show that we did have contamination in our experiment. In our third round of qPCR, the negative control was 31.15 in bacteroidetes, and we didn't get a negative control for a varicue microbia. Again, we had a high negative control here, which is good, but we had values that were close to or exceeded the negative control. We got values of 31.06, 34.12, 26.07, and 21.12. And since the negative control was 31.15, and we got a value of 31.06 and 34.12, these are both really high and really close to our negative control, and one that even exceeded it. So we did have contamination here. As for our hypothesis, we did not find any data to support our hypothesis. So we originally hypothesized that there would be differences in the gut microbiomes of dogs with epilepsy and dogs with lowered kidney function. We thought that there would be similarities in the microbiome of the dogs with the same health problems, but we were unable to find a correlation between any of the gut microbiomes of any of these dogs. So the kidney disease dogs didn't have a similar gut microbiome, the epilepsy dogs didn't have a similar gut microbiome, and none of them had a similar gut microbiome to each other. So our hypothesis was completely wrong. So in the future, we decided we wanted to perform next-gen analysis, which would allow us to look at all of the bacteria in all of the samples to potentially find a correlation between the samples, because we do believe that there could be a correlation between health problems and gut microbiome. But since we weren't able to find any data, we might have to change our hypothesis in the future to make it something that we can find data to support. But first, we want to try the next-gen analysis to see if we can find any data to support our hypothesis. That's all for the Scout Microbiome project. Thanks for watching.